Yes, so my name is Erica Goss, and I'm a professor at the University of Florida in Gainesville, Florida. And today I'm going to talk about copper resistance in the bacterial spot pathogen Xanthomonas euvesicatoria pathovar perforans, which I may call perforans or XEP. And I'm talking about this today because copper is really important, has been very important for managing bacterial spot and other bacterial diseases. But the pathogen can respond in turn. And what we've learned about the evolution of copper tolerance in this pathogen, I think, can be applied more broadly in thinking about why this pathogen is such a problem and how we can deal with the rapidly changing population in the field. So first, I just want to acknowledge that this is really a collaborative effort, particularly with Jeff Jones and Gary Balad, but also other faculty and postdocs and students and many collaborators who have supported this research in various ways. And a lot of the work I'm going to talk about today was done primarily by Amandeep Kaur, who was a former postdoc with us. So in Xanthomonas perforans, we've seen rapid change in the population since it was first discovered in 1991. So its first isolation was from tomato in Florida in 1991, and by 10, 20, or 15 years later, there was complete replacement of the previous bacterial spot population on tomato, which was Xanthomonas euvesicatoria, with Xanthomonas perforans, or XEP. And at first, XEP was sensitive to copper-based products. So this was actually kind of a relief. It was able to be controlled, whereas Xanthomonas euvesicatoria had been resistant or tolerant to copper. But in the recent years, we've seen increasingly aggressive disease. The population overcame the only resistance available in tomato. And as I'm going to talk about today, we've seen nearly 100% copper tolerance. And in the last seminar, Gary Balad sort of gave the history on that increasing copper tolerance here in Florida, which has also been seen in other regions, for example, in the Midwest. We also are now seeing Xanthomonas perforans on pepper, so an emerging population there. So to start, I want to briefly talk about the changes we've seen in terms of the aggressiveness of the disease to show how this pathogen is evolving rapidly over time, and then we'll transition into copper. So what we noticed in samples from 1991 through 2017 was increasing frequency of this virulence gene called ZOPJ2 or AVRBST. And so because we were seeing that increase in frequency, in fact, it wasn't in the bacterial population at all in 1991, we decided to investigate what the effect this gene was having in Xanthomonas perforans. And so a series of experiments were done using strains that had the gene and strains that were equivalent in every way except the ZOPJ2 gene was deleted. And you can see in these leaflets that there are more lesions and more severe disease in the same strain with and without this gene. So a lot of work went into experiments in the field and subsequent modeling, which showed that the rate of spread of perforans in the field is 3 times faster when the gene is present. So this was based on experiments and modeling. And so these are modeling results showing if you have one infected plant in the middle of a field, what you might expect to see at the end of the season with the ZOPJ2 gene versus without the ZOPJ2 gene. So this is just one example of how we've seen genes move into this pathogen that make it more aggressive. And I wanted to show this because it's an example of horizontal gene transfer, which is relevant for copper resistance as well. So what is horizontal gene transfer? Bacteria have their chromosomal DNA, which they inherit just as we inherit our DNA from one generation to the next. But they also have these small pieces of DNA, smaller pieces of DNA called plasmids that can move from one bacterial cell to another by conjugation. And so what can happen here is that a donor cell can have a plasmid and then transfer it to another cell that has a different genetic background, so has a different set of genes in the chromosome. And now this cell has that piece of DNA with containing potentially lots of genes as well. So this is a way that bacteria can rapidly spread genes in a population. And these genes tend to be coding for important traits. For example, virulence genes like ZOPJ2, as well as antimicrobial resistance genes and heavy metal resistance genes. So this is a mechanism that can be used by bacteria to rapidly spread a new trait across populations. And so the copper resistance trait in Xanthomonas is known to be carried on such a plasmid. And the COP genes are a cluster of genes that confer copper tolerance. And I'll use copper resistance and copper tolerance somewhat interchangeably. Copper tolerance is sort of more correct in that we know that there's increasing tolerance, that it confers increasing quantitative tolerance, but that there is a limit to the resistance, right? The tolerance trait. So it's not all or nothing. But these are called copper resistance genes, so I'll use that term as well. So the COP genes that confer copper resistance are on this pretty large plasmid in Xanthomonas, and this is known from multiple other species as well as other bacterial spot pathogens that these genes are on a 230 kilobase pair plasmid. So that's pretty big. We more recently learned that these COP genes, these copper resistance genes, are also on the chromosome in Xanthomonas euvesicatoria pathovar perforans. And they're on a chromosome in what's called a genomic island. And so this is called a genomic island because it's a piece of DNA that has been inserted into the chromosome and has different characteristics than the rest of the chromosomal DNA. So that sort of makes it an island. And these genomic islands are also thought to be horizontally transferred either from plasmid or from phage. So we already know that copper resistance is a trait that is acquired by horizontal gene transfer. Our question was, which is more common in XEP? Are the copper genes, copper resistance genes on the plasmid or the chromosome in these populations? And why we want to know that is because it can potentially affect the expression of that trait. So it can affect the level of copper tolerance that they exhibit, and I'll explain why that is in a few minutes. And it also may affect the persistence of copper tolerance in the population, because the conventional wisdom is that this large plasmid could be lost from the bacterial population. It could be lost from these cells because it's large and maybe difficult to maintain, But once the genes are in the chromosome, they may be less likely to be lost because the bacteria has to retain the chromosome and the genes on them to survive. So we took a large sample of genomes of XEP and examined them for the COP genes. So we had 452 genomes that we looked at, 427 from the US, and most of those were from Florida. And we found that only 27 of those genomes had no copper resistance genes at all. So only 6% had no genes and is expected to be sensitive to copper. The copper resistance genes were on the genomic island on the chromosome in 264 genomes. So that's the majority of strains had those genes already on the chromosome. So there's our little genomic island. 100 of the strains had that large plasmid with the COP genes. So that's about 22% of strains. Well, we were surprised to see that there was also a much smaller plasmid, about a third of the size, with the COP genes. And we saw that in 59 strains, or about 13% of our sample. So when we investigated that further to make sure that we were actually seeing a small plasmid and not just a piece of the large plasmid, we found that those strains with that smaller plasmid also have the COP genes on the chromosome. So these strains have 2 copies of the copper resistance gene cluster on a plasmid and on a chromosome. And finally, what we observed was that all US strains had identical nucleotide sequences for the COP genes. And this was surprising to us because we think of copper resistance being acquired by horizontal gene transfer from wherever the bacteria can get it, and that might be in different environments and might lead to differences in the sequences of the COP genes. But in fact, we saw that strains from Florida and Ohio and Alabama all had the identical genes. And it didn't matter if they were in the chromosome, on the large plasmid, or the small plasmid. So this suggests that these genes spread very rapidly throughout the population in the US. And in fact, when we looked at strains from other countries, they did have differences in the COP genes in the nucleotide sequence, as we would expect. So where did this 87 kb plasmid come from? Well, we think it's the result of recombination between this large plasmid and another very small plasmid. So this piece of the large plasmid with the copper resistance genes is still in this small plasmid, but also contains a piece of another plasmid that's in Xanthomonas perforans. And so what this pathogen is doing is keeping the genes it needs and discarding the genes that presumably it doesn't need from this large plasmid. We can also look at the context for these plasmids, the genomic context. So what we did was look at all these strains. This was a different group of strains from Florida, but we looked at the plasmid diversity in the strains. And what you're seeing here is a plasmid network. So these bursts that look like fireworks are a particular plasmid in a bunch of different strains. And the gray lines between these bursts indicate some shared gene content between plasmids. But what is evident here is that we have 2 different groups of plasmids, one in these blue ellipses and a separate group of plasmid in the green ellipse here. And this is indicating genetic groups based on the chromosomal DNA. So as Gary Vlad briefly explained in the last seminar, we classify the genetic diversity in XEP based on genes in the chromosome. And so we have clusters 3 and 6 here, and clusters 3 and 6, based on the chromosomal DNA, tend to have this group of plasmids, whereas the other clusters we see in the Florida population have these other plasmids. So what we think happened is that the large plasmid with the copper resistance genes was not compatible in this genomic background for some reason, but the recombination between the large plasmid and the other small plasmid that created the 87 kb plasmid allowed this plasmid to persist, to be able to spread in this population of the pathogen. All right, so what does all this mean for copper tolerance? So what we did was measure copper tolerance in vitro in the lab on media containing copper sulfate at different concentrations. So this is an original 1991 strain. As I mentioned, it was copper sensitive, so it will grow, spots of the cells will grow on media with no copper sulfate, but at higher concentrations, there's no growth. So to compare this to a strain that has the copper genes in the chromosome, now we see growth with no copper sulfate. We see growth at 200 ppm, which is our sort of threshold for calling a strain copper resistant. And we see growth at higher concentrations of cells, at much higher concentrations of copper sulfate. So even if we have enough cells, we can even see growth at 350 ppm. When we look at the copper genes in the large plasmid, we see even higher growth. So we are more growth at higher concentrations of copper sulfate. So we see growth at all concentrations of the spots of cells at 250, 300, and even up to 400 ppm. Finally, what if they have copies on the small plasmid and the chromosome? We see even more growth at these highest concentrations of copper. So strains carrying the COP genes on plasmids are more tolerant to copper in vitro than strains with the chromosomal COP genes. And if you have more copies, even the better. And so here we're kind of assuming that the plasmid may confer more copper tolerance because it has more copies of the gene, because the plasmid can be present in the cell in multiple copies, but we wanted to test that. So we did that with qPCR of one of the genes, the copper genes in this copper gene cluster, and we compared the number of copies of that gene in a strain with it only on the chromosome, in a strain with the large plasmid, and in a strain with the chromosome and the small plasmid. So what we see is that that large plasmid is present in cells in maybe one or two copies. But when you have the small plasmid, you get 9 times more copies of the COP-B gene, so these copper resistance genes, than if you only have it, if the bacteria only has it on the chromosome. So how does this compare to gene expression? So here we're actually quantifying the expression, the transcription of the gene, the copper resistance gene, and we're setting the expression level of the strain with just the copy on the chromosome to 1. And what we see that is, if in the strain with the large plasmid, we see 10 times higher expression. And in the strain with the small plasmid, we see many hundreds times higher gene expression of this copper resistance gene than compared to if it was in the chromosome alone. So we think that higher gene copy number and much higher gene expression is conferring higher tolerance to copper when there are multiple copies of the gene. When it's in the plasmid, and that it may depend also on the size of the plasmid. So our assumption was that this large copper plasmid is unstable because it's large and it has to be maintained by these cells. And so we then wanted to test this. So what we did was grow strains with the large plasmid in the lab without copper. So if there's not selection from copper, will these strains maintain the large plasmid? So we maintain them in liquid media for 14 days, transferred daily, and we estimate that we went through about 160 generations of growth without selection from copper. Then screened hundreds of resulting single colonies from the final generation on media with and without copper sulfate. So we were looking for colonies that only grew in media without copper, meaning it had lost that large copper plasmid. So what we found was that very few strains lost the large plasmid in the absence of copper. So 3% or less of the resulting hundreds of colonies we examined had lost the large plasmid and had lost copper tolerance. And the frequency of loss of the large plasmid depended on the genetic background of the strain. So cluster 6 strains had some loss of the large plasmid, whereas in all the many hundreds of strains, only one strain lost the large plasmid in cluster 3. So this suggests that even this large plasmid is fairly stable in the population. So in summary, we found evidence for strong selection for copper tolerance in Xanthomonas perforans in multiple ways that it has responded to this strong selection in terms of the evolution of its genome and the copper resistance genes in the genome. So first we found that there was identity of the genes across all strains in the US, suggesting that this trait, these genes, spread rapidly in the population. We saw integration of the genes in the chromosome, which we believe will make it hard for the population to lose this trait. So they'll retain, the bacterial will retain copper tolerance. And we saw recombination of plasmids with these COP genes, which shows the flexibility of this bacteria and the different strategies it will use to allow spread of a beneficial trait when there is selection for that trait. Unfortunately, we did not see that the large plasmid was easily lost even in the absence of copper, and this went against what we had recently observed in Xanthomonas euvesicatoria on pepper, where we saw that the strains we collected from a farm that didn't use copper to manage the disease were all copper sensitive. For Xanthomonas perforans on tomato, it doesn't seem that this is likely to happen. Copper tolerance seems like it's going to persist in the population. So this work is sort of an example, is illustrative of how Xanthomonas perforans responds to our efforts to manage it. So seems like it can evolve very quickly and use multiple different genetic strategies to respond to selection from management. And this is something to keep in mind as we develop new management approaches and need to watch for how it may respond to overcome those, which may happen quickly depending on the tools it has available to use in terms of overcoming resistance or overcoming chemical management. We are also, as part of this TomSpot project, categorizing the variation in this pathogen. So I talked about the clusters, I talked about the variation in the plasmids in this population. And all of this is going towards being able to recognize aggressive variants and the characteristics, the genetic characteristics of aggressive variants, so that we can rapidly detect those and see when the population is yet again changing and we need to respond to that. So thank you for joining today, and I'm happy to take questions. If you're interested in the papers that I talked about today, here's some links to those papers.